Hello, welcome to World of Microbes. This will be a detailed video of mycology. In this video we will discuss about the characteristics of fungi, everyday impact of fungi, common types of fungi, understanding the structure of fungi, the classifications and useful properties of fungi. A brief video of introduction to mycology is also uploaded in the channel. The link is in the description. Please do like and subscribe. Mycology is the study of fungi, including mushrooms, molds, and yeasts. Fungi are unique life forms that have specialized eukaryotic cells. This means that most fungi are multicellular organisms that have an organized nucleus. However, some fungi are single-celled organisms. The study of fungi is useful for many reasons. They play significant roles in ecosystems, often acting as decomposers that break down organic matter, and some species are known for their medicinal properties, such as the production of antibiotics like penicillin. Mycology also addresses the harmful aspects of fungi, as certain species can cause diseases in plants and humans, highlighting the importance of research in this field for both health and agriculture. Mycologists study these dangerous organisms to try to find ways to prevent or destroy them. Mycology was at first not a separate discipline, but was part of botany, dealing only with the detailed study of fungi. It was not until the 19th century, when the unique characteristics and importance of fungi in nature and their impact on various aspects of human life were understood, that mycology began to develop as an independent science. It became a key discipline in ecological, medical, industrial, and agricultural research, offering a wide range of knowledge about the diversity, functions, and uses of fungi. Today, mycologists not only identify and classify different fungal species, but also study their genetics, biochemistry, and role in ecosystems, contributing to innovative discoveries in many industries. It is worth mentioning that the term mycology was also used in Poland, which has been replaced by the currently used term mycology. Before mycology became a rigorous modern science, ancient cultures viewed mushrooms with immense fascination, treating them as botanical anomalies, divine foods, or dangerous omens. Because fungi sprout rapidly, thrive in the dark, and include hallucinogenic and deadly varieties, they were wrapped in intense folklore. A look at early observations and myths reveals how our ancestors interacted with the fungi kingdom. 1. Divine foods and immortality. Ancient Egypt, mushrooms were considered sons of the gods and plants of immortality. The Egyptians believed that the storm god Set hurled lightning bolts that seeded mushrooms to the earth. Consuming them was an exclusive privilege reserved for pharaohs, while commoners were forbidden to even touch them. Mesoamerica, indigenous cultures, such as the Aztecs, referred to psychoactive mushrooms, like psilocybe, as tiananacatl, which translates to flesh of the gods. They were revered as bridges between the natural world and the divine, used by shamans to communicate with the spiritual realm. 2. Early scientific classifications. Ancient Greece. The Greek philosopher Theophrastos, 371 to 288 BC, was one of the first to attempt to classify plants systematically. However, because fungi lack traditional roots or leaves, he grouped them as plants missing certain organs. Roman era. Pliny the Elder observed truffles and other fungi in his Natural History. He inaccurately deduced that fungi were simply superfluous moisture of earth, rotten wood, and of other rotting things. This idea that mushrooms were born from decay dominated European understanding for centuries. 3. Witches, devils, and fairy rings. During the Middle Ages in Europe, mushrooms gained a deeply dark reputation. Lacking scientific understanding, people associated fungi with the macabre and evil. Fairy rings, naturally occurring circular arcs of mushrooms, were seen as bewitched traps where witches gathered to dance on Walpurgis night. People avoided walking into them and warned their livestock away, believing it would taint the cow's milk. Witches' butter and devil's fingers, creepy or bioluminescent fungi, were attributed to spells. Fungi like the yellow brain were called witch's butter. Finding one on your doorstep was interpreted as a curse. The only way to break the spell was to stick pins into the fungus until it disappeared. 4. Bizarre rules and superstitions. Without microscopes or chemical analysis, foragers and classical naturalists developed elaborate, often fatal myths to distinguish edible mushrooms from poisonous ones. The iron myth. Ancients believed poisonous mushrooms were caused by rusted or dissolved iron. Some believed that dangerous mushrooms only grew near the dens of venomous beasts, rotten clothes, or dung. The silver spoon test. A long-standing superstition held that cooking wild mushrooms with a silver spoon would reveal their toxicity. If the silver turned black, the mushroom was deadly. The color test. A surviving, dangerous misconception is that mushrooms turning blue when touched or exposed to air are automatically toxic. While some poisonous mushrooms blue, many edible, sought-after species, like the Boletus edulis family, also bruise blue. The dawn of scientific mycology marks the transition of fungal studies from ancient folklore to a rigorous biological discipline. Once lumped into botany, fungi gained their own scientific independence and identity, driven by early microscopic discoveries and detailed morphological classification. Pier Antonio Micheli, 1729, widely considered the father of modern mycology, this Italian botanist illustrated over 900 species in his landmark work, Nova Plantarum Genera. He was one of the first to observe fungal spores under a microscope and recognized their role in reproduction. Heinrich Anton de Berry, 1850s-1880s, often hailed as the founder of modern mycology and plant pathology, this German surgeon and botanist famously elucidated the life cycle of Phytophthora infestans, proving it was the fungal pathogen responsible for the potato late blight. Christian Hendrik Persoon and Elias Magnus Fries, these botanists laid the crucial groundwork for modern fungal taxonomy by establishing standardized systems for naming and classifying fungal species. For centuries, fungi were grouped with plants because of their stationary nature and growth habits. However, as scientific tools advanced, mycologists realized fungi lacked chlorophyll and were actually evolutionarily more closely related to animals than plants. The pathogen era refers to a growing shift in mycology towards studying fungi as deadly infectious agents rather than just ecological recyclers. Concurrently, independence marks mycology's move to establish itself as a standalone scientific discipline, distinct from botany and general microbiology. The pathogen era. Modern mycology has entered an age where fungal diseases pose catastrophic threats to global health, agriculture, and wildlife. Clinical rise. Opportunistic fungal pathogens, such as Candida auris, Aspergillus, and Cryptococcus, are causing life-threatening systemic infections, heavily driven by an expanding population of immunocompromised patients. Environmental devastation. Fungal pathogens are wreaking havoc on ecosystems. For instance, chytrid fungi are driving worldwide amphibian declines, while white nose syndrome is decimating bat populations. Agricultural threats. Phytopathogenic fungi cost billions annually by devastating staple crops, like maize and rice. This has triggered massive research into fungal effectors, mycotoxins, and novel antifungal treatments. Independence in mycology. Historically treated as an offshoot of botany, since fungi were originally classified as plants, mycology has continually fought for its own taxonomic and academic autonomy. Overcoming the holomorph problem. For decades, sexual stages, teleomorphs, and asexual stages, anomorphs, of the same fungus were classified as completely independent species. Recent revisions by the International Botanical Congress established the one fungus, one name principle, giving mycology a rigorous, independent naming system. Culture-independent diagnosis. Mycologists rely less on slow, traditional culturing methods and more on rapid, molecular tools like PCR and genomic sequencing. This allows for exact identification of fungal pathogens without needing to isolate them in a lab first. Modern mycology merges classical taxonomy with advanced molecular biology and genomics to explore the immense potential of fungi. Fungal biotechnology leverages these organisms to create sustainable biomaterials, life-saving pharmaceuticals, and agricultural solutions, driving innovations that replace petroleum-based plastics and improve global health. Key pillars of fungal biotechnology. Sustainable biomaterials. The root structures of fungi, mycelium, are engineered into eco-friendly packaging, structural insulation, and durable mycoleather to reduce industrial waste. Pharmaceuticals and medicine. Fungi remain biological powerhouses for synthesizing life-saving antibiotics, e.g., penicillin, and immunosuppressants, while modern mycology investigates new fungal metabolites for antiviral and antimalarial treatments. Bioremediation. Biotechnologists utilize specialized fungal strains capable of breaking down environmental toxins and degrading tough plastics, like polyurethane, into harmless byproducts. Food and agriculture. Fungi are indispensable in alternative protein production, enzyme manufacturing, and biofertilizers that enhance soil health and crop resilience without harsh chemical additives. Modern techniques transforming the field. Metabolic engineering, modifying fungal genomes to hyperproduce specific, high-value enzymes, organic acids, and biofuels. CRISPR-Cas9 editing, allowing precise modification of fungal DNA to optimize industrial fermentation yields and discover hidden biosynthetic pathways. Mycology is the branch of biology dedicated to the study of fungi, including their genetic, biochemical properties, taxonomy, and ecological impact. It is divided into several specialized subdisciplines based on the specific applications, environments, or host organisms of the fungi being studied. Applied mycology is the practical branch of biology that focuses on leveraging fungi for human benefit, environmental sustainability, and industrial advancement. Rather than just observing or classifying fungal species, applied mycologists study how to utilize yeasts, molds, and mushrooms to solve real-world problems across diverse sectors. Fungi serve as highly efficient biological engines because of their unique genetics, rapid growth, and powerful enzymatic properties. Medicine and pharmaceuticals. Fungi are a foundational pillar of modern healthcare and clinical therapies. Antibiotics, yielding life-saving drugs like penicillin, streptomycin, and tetracycline. Statins, producing critical biological compounds used globally to lower blood cholesterol. Immunosuppressants, sourcing complex fungal metabolites crucial for preventing organ transplant rejection. Myconanotechnology, engineering metal nanoparticles synthesized by fungi to target and manage dangerous pathogens. Food and agriculture. Fungal interactions are essential for maintaining global food security and supply chains. Fermentation, powering long-established baking, brewing, dairy, and wine processing industries using yeasts. Biofertilizers, employing symbiotic mycorrhizal and endophytic fungi to boost crop growth and soil fertility. Biopesticides, deploying specific fungal strains as safe, non-chemical bioherbicides and disease suppressors. Mycoprotein, cultivating fungal biomass to create highly nutritious, meat alternative protein sources. Mycotoxin management, tracking and neutralizing toxic secondary metabolites that contaminate global grains. Industrial biotechnology and materials. Fungi are driving a massive shift toward eco-friendly manufacturing and a circular economy. Enzyme production, harvesting fungal enzymes for commercial textiles, detergents, and industrial chemical processing. Mycomaterials, growing fungal mycelium into biodegradable packaging, eco-leather, insulation, and high-strength bricks. Biofuels, utilizing specialized wood decay strains to break down tough cellulose into clean hydrocarbons and biofuels. Ecology and environmental management. Fungal networks are highly adaptive tools used to restore damaged natural ecosystems. Micromediation, using fungal species to clean up landfills, degrade toxic azo dyes, and metabolize hazardous chemical waste. Building mycology, assessing how molds and structural fungi degrade building materials to prevent property damage and health issues. Medical mycology is the study of fungi that cause diseases in humans and animals. It focuses on identifying fungal pathogens, diagnosing invasive infections, mycoses, and developing treatments. With rising rates of immune suppression globally, it is a critical and growing branch of clinical microbiology. Fungal diseases range from mild, superficial skin rashes to life-threatening systemic infections. The most clinically significant categories include yeasts, single-celled fungi like Candida, which causes thrush, and Cryptococcus, which can cause severe meningitis. Molds, filamentous fungi like Aspergillus and Mucorales, which are common in the environment, but can cause severe pulmonary and invasive infections in immunocompromised patients. Thermally dimorphic fungi, fungi that change forms based on temperature. They include histoplasma and coccidioids, which typically cause respiratory illnesses. Types of fungal infections. Medical mycology categorizes these conditions based on the depth of tissue invasion. Superficial or cutaneous, affects the skin, hair, and nails, e.g., ringworm, athlete's foot, and mycosis. Subcutaneous, infections of the deeper layers of skin and muscle, often resulting from traumatic inoculation, e.g., sporotrichosis. Systemic and invasive, life-threatening infections that spread through the bloodstream to internal organs, often primarily affecting immunocompromised hosts. Systematic mycology is the branch of biology dedicated to the classification, identification, and evolutionary tracing of fungi. It maps fungal diversity, establishes species relationships using genetic and morphological data, and is essential for understanding fungal evolution, ecological roles, and applications in medicine and biotechnology. Core disciplines: taxonomy, naming, describing, and classifying fungi based on morphological traits and genetic relatedness. Phylogenetics, using molecular data, DNA sequences, to determine evolutionary lineages and construct fungal family trees. Industrial mycology is the branch of applied biotechnology that exploits fungi, including yeasts, molds, and mushrooms, to manufacture commercial products. It utilizes the natural metabolic diversity of fungi to scale up the production of medicines, foods, industrial enzymes, and environmental solutions. Pharmaceuticals and medicine. Fungi are prolific producers of secondary metabolites used to create life-saving drugs. Antibiotics. Penicillium chrysogenum is used to mass-produce penicillin, while other fungi yield cephalosporins. Immunosuppressants. Cyclosporin A, derived from Tolypocladium inflatum, is critical for preventing organ transplant rejection. Statins. Lovastatin, produced by Aspergillus terreus, serves as a frontline treatment to lower blood cholesterol. Anticancer agents. Fungal systems are continually screened to discover novel tumor-inhibiting compounds. Food and beverage production. Fungi drive both traditional fermentations and modern alternative protein industries. Fermentation. Saccharomyces cerevisiae, bakers' or brewers' yeast, powers the baking, beer, and wine sectors. Organic acids. Aspergillus niger is heavily utilized to produce citric acid and gluconic acid for food preservation. Mycoprotein. Species like Fusarium venenatum are grown in large bioreactors to produce meat alternatives like corn. Solid-state products. Molds drive the production of traditional foods like soy sauce, tempeh, and various cheeses. Industrial enzymes and biofuels. Fungal enzymes break down complex polymers, replacing harsh chemicals in manufacturing. Textiles and detergents. Fungal cellulases, proteases, and amylases fade denim, soften fabrics, and remove laundry stains. Paper and pulp. Xylonases and lignin-degrading enzymes from white rot fungi bleach paper pulp sustainably. Biofuels. Fungi break down agricultural biomass into fermentable sugars to generate bioethanol. Agriculture and bioremediation. Industrial mycology provides eco-friendly alternatives to chemical fertilizers and pest control. Mycorrhizal inoculants. Commercial fungi form symbiotic relationships with plant roots to enhance nutrient uptake. Biopesticides. Entomopathogenic fungi act as natural killers against agricultural insect pests. Micromediation. Fungal mycelium absorbs and breaks down heavy metals, plastics, and petroleum hydrocarbons from contaminated soils. Agricultural mycology is the study of fungi and fungal-like organisms that impact agricultural crops, livestock, and soils. It is a critical scientific field focused on mitigating crop diseases, and harnessing beneficial fungi to enhance sustainable food production and soil health. The discipline is primarily divided into two main areas based on the relationship between fungi and agricultural systems. 1. Plant pathology. The harmful, many fungi act as plant pathogens that cause devastating crop diseases and significant yield losses. Agricultural mycologists study the life cycles, genetics, and spread of these fungi to develop effective management strategies. Common pathogens includes blights, rusts, mildews, and molds, e.g., Fusarium wilt, Botrytis gray mold. Mycotoxins. Some fungi produce toxic compounds in crops, like Aspergillus producing aflatoxin, posing serious threats to food safety and livestock health. Management focuses on developing disease-resistant crop varieties, applying biofungicides, and implementing careful post-harvest storage techniques. 2. Beneficial fungi. The helpful. A wide variety of fungi actively promote agricultural productivity and plant health. Agricultural mycologists harness these to create eco-friendly alternatives to chemical fertilizers and pesticides. Mycorrhizal fungi form symbiotic relationships with plant roots, expanding their root networks to vastly improve water and nutrient absorption, e.g., phosphorus. Biofertilizers. Certain fungi increase soil fertility and help cycle vital nutrients. Biopesticides and biological control. Entomopathogenic fungi, like Beauveria bassiana, naturally infect and control crop pests, while nematophagous fungi target crop-eating nematodes. Building mycology is the study of fungi within the built environment. It focuses on the identification, management, and prevention of fungi, such as mold and wood-decaying organisms, that degrade construction materials and pose health risks to building occupants. Key concepts in building mycology. Biodeterioration. Fungi feed on organic construction materials, e.g., timber, drywall, wallpaper. Wood-decaying fungi, dry rot, wet rot, are especially destructive to structural integrity. Indoor air quality. Mold growth releases spores and mycotoxins into the air, which can trigger allergies, respiratory issues, and other health hazards. Microclimates. Fungi thrive in damp, stagnant environments. Building mycology analyzes how poor ventilation, moisture ingress, and thermal bridging create ideal ecological niches for fungal settlement. Common fungi in buildings. Aspergillus and Penicillium, common indoor molds that can cause respiratory irritation and allergic reactions. Stachybotrys chartarum, black mold known for producing hazardous mycotoxins, typically found on water-damaged cellulose materials like drywall. Serpula lacrimans, dry rot, the most notorious wood-destroying fungus in buildings, capable of moving through masonry to find new wood sources. Management and control strategies. Moisture control. The most effective defense against fungal growth is keeping building interiors dry. This involves fixing leaks, ensuring proper drainage, and maintaining humidity levels below 50 to 60%. Ventilation. Proper HVAC design and mechanical ventilation prevent the accumulation of stagnant, moisture-laden air. Inspection and remediation. Professionals use culture-based methods, moisture meters, and air sampling to assess mold contamination and implement remediation strategies. Veterinary mycology is the study of fungi, including yeasts, molds, and dimorphic fungi, that cause infections or produce toxins in animals. It covers domestic companion animals, livestock, poultry, and wildlife. Because many animal-associated fungi are zoonotic, can jump from animals to humans, this field forms a critical bridge under the One Health approach. Classification of animal mycoses. Fungal infections in veterinary medicine are traditionally categorized based on the depth and tissue type they invade. Superficial or cutaneous mycoses. These affect the dead layers of skin, hair, and claws. The most common are dermatophytoses, universally known as ringworm, caused by species of Microsporum and Trichophyton. Yeast-related conditions like Malassezia pachydermattis cause severe ear infections, otitis, and greasy skin inflammation in dogs. Subcutaneous mycoses. These stem from environmental trauma that introduces fungi directly beneath the skin. For example, sporotrichosis, Sporothrix species, forms draining nodules in outdoor cats and is highly infectious to humans. Systemic mycoses. These are deep-seated, often fatal infections affecting major internal organs like lungs, bones, and the nervous system. Many are caused by dimorphic fungi, which live as molds in the environment, but morph into pathogenic yeasts at animal body temperatures. Classic examples include blastomyces, histoplasma, and coccidioids. Opportunistic mycoses. These occur when a standard, benign fungus takes advantage of a compromised immune system. Examples include aspergillosis in the respiratory tracts of birds or dogs, and candidiasis overgrowths. Biology and characteristics. Fungi hold unique properties that distinguish them directly from bacteria. Eukaryotic cells, they have a membrane-bound nucleus and organelles. This means they share structural similarities with animal host cells, making targeted drug therapies challenging to design without toxicity. Ergosterol, their cell membranes contain ergosterol instead of cholesterol. This serves as the primary target for modern veterinary triazole antifungals. Chitinous walls. Fungal cell walls are structurally rigid, composed primarily of chitin and glucan polymers. Antibiotic insensitivity. Fungi are completely unaffected by conventional antibacterial drugs. Misusing antibiotics can actually wipe out competitive bacteria, leading to secondary fungal overgrowths. Emerging threats. The landscape of veterinary mycology is changing rapidly due to several global influences. Antifungal resistance. The excessive use of agricultural fungicides shares structural classes with veterinary medicines, accelerating resistant strains in companion animals and livestock. Climate change. Shifts in regional temperatures and humidity are altering the geographic range of soil-dwelling fungi, pushing deep systemic mycoses into areas where they were historically unseen. Biodiversity loss. Fungal pathogens can cause massive die-offs in fragile wildlife populations, notably affecting amphibians and marine species. Fungi are a diverse group of eukaryotic organisms, including mushrooms, molds, and yeasts, that digest food externally and absorb nutrients directly. Unlike plants, they cannot make their own food. Genetically, they are actually more closely related to animals than to plants. Fungus. Any of about 144,000 known species of organisms of the kingdom fungi, which includes the yeasts, rusts, smuts, mildews, molds, and mushrooms. There are also many fungus-like organisms, including slime molds and oomycetes, water molds, that do not belong to kingdom fungi, but are often called fungi. Many of these fungus-like organisms are included in the kingdom Chromista. Fungi are among the most widely distributed organisms on Earth and are of great environmental and medical importance. Many fungi are free-living in soil or water. Others form parasitic or symbiotic relationships with plants or animals. Fungi are eukaryotic organisms, i.e., their cells contain membrane-bound organelles and clearly defined nuclei. Historically, fungi were included in the plant kingdom, however, because fungi lack chlorophyll and are distinguished by unique structural and physiological features, i.e., components of the cell wall and cell membrane, they have been separated from plants. In addition, fungi are clearly distinguished from all other living organisms, including animals, by their principal modes of vegetative growth and nutrient intake. Fungi grow from the tips of filaments, hyphae, that make up the bodies of the organisms, mycelia, and they digest organic matter externally before absorbing it into their mycelia. While mushrooms and toadstools, poisonous mushrooms, are by no means the most numerous or economically significant fungi, they are the most easily recognized. The Latin word for mushroom, fungus, plural fungi, has come to stand for the whole group. Similarly, the study of fungi is known as mycology, a broad application of the Greek word for mushroom, myx. Fungi other than mushrooms are sometimes collectively called molds, although this term is better restricted to fungi of the sort represented by bread mold. A fungus is a eukaryote that digests food externally and absorbs nutrients directly through its cell walls. Most fungi reproduce by spores and have a body, thallus, composed of microscopic tubular cells called hyphae. Fungi are heterotrophs and, like animals, obtain their carbon and energy from other organisms. Some fungi obtain their nutrients from a living host, plant or animal, and are called biotrophs. Others obtain their nutrients from dead plants or animals and are called saprotrophs, saprophytes, saprobes. Some fungi infect a living host, but kill host cells in order to obtain their nutrients. These are called necrotrophs. Fungi were once considered to be primitive members of the plant kingdom, just slightly more advanced than bacteria. We now know that fungi are not primitive at all. In fact, recent taxonomic treatments such as the Tree of Life project show that fungi and animals both belong to the group Opisthokonta. Fungi may not be our next of kin, but they are more closely related to animals than they are to plants. We also recognize that organisms traditionally studied as fungi belong to three very different unrelated groups: the true fungi in kingdom fungi, Eumycota, the oomycetes, and the slime molds. Let's briefly consider the major groups in kingdom fungi. They will be described in greater detail in later videos. Open most introductory mycology books and you'll see that there are four main groups, phyla, of true fungi: Ascomycota, Basidiomycota, Chytridiomycota, and Zygomycota. Recent studies have provided support for the recognition of additional phyla, such as Glomeromycota, a group of fungi once placed in Zygomycota that form an association with the roots of most plants. A group of parasitic organisms called Microsporidia that live inside the cells of animals are also now considered to belong in the fungal kingdom. Hibbert et al., 2007, published a comprehensive classification of the kingdom fungi, the result of collaboration among many fungal taxonomists. This classification is used in the Dictionary of the Fungi, Kirk et al. 2008, and other fungal references and databases. However, the classification system will undergo additional changes as scientists use new methods to study the fungi. For example, Jones et al. 2011 described the Cryptomycota, a potentially new phylum of organisms within the kingdom fungi. Fungi are among the oldest life forms on Earth, with evolutionary origins stretching back 1 to 1.4 billion years ago. This means they conquered the Earth's surface hundreds of millions of years before the first plants or animals appeared. The timeline of fungal evolution breaks down as follows. Ancient origins 1 to 1.4 billion years ago. Genetic and molecular clock analyses suggest that the earliest single-celled, aquatic ancestors of modern fungi diverged from the lineage that would eventually lead to animals. Deep-sea microfossils. 2.4 billion years ago, some scientists have discovered tantalizing, mycelium-like microfossils in deep-sea rocks in South Africa. If confirmed as true fungi, this pushes their origins back by another billion years, suggesting they evolved in the deep ocean. Conquering land. 635 to 440 million years ago. The oldest definitive land fungus fossils date back roughly 635 million years. And a 440 million year old microfossil, a Tortotubus, represents some of the oldest complex life found on terrestrial land. Fungi were vital in shaping the planet's first soils and ecosystems, acting as the primary decomposers that paved the way for land plants. Fungi are eukaryotic, spore-producing organisms that lack chlorophyll. Unlike plants or animals, they are heterotrophs that excrete digestive enzymes externally, absorbing nutrients directly from their environment. Cellular and structural traits. Eukaryotic. Fungal cells contain membrane-bound organelles, including a well-defined nucleus and mitochondria. Rigid cell walls. Unlike plants, which use cellulose, fungal cell walls are primarily composed of chitin, the same tough polymer found in the exoskeletons of insects. Cell membranes. Their membranes are stabilized by ergosterol, a steroid molecule that replaces the cholesterol found in animal cells. Lack of chlorophyll. Fungi cannot photosynthesize and do not contain chloroplasts. Nutrition and metabolism. Heterotrophic by absorption, fungi survive as decomposers, saprobes, parasites, or mutualists. They release powerful enzymes to break down organic matter in their surroundings, absorbing the dissolved nutrients. Energy storage. Similar to animals, fungi store their excess carbohydrates as glycogen, rather than starch. Body structure. Filamentous networks. Hyphae. The vegetative body of most fungi is a tangled mass of slender, thread-like filaments called hyphae. Mycelium. This collective network of hyphae forms the thallus, body, of the fungus, allowing it to grow deep into a food source. Unicellular exceptions. Some fungi, such as common yeasts, do not form hyphae and exist as single-celled organisms. Reproduction. Spore formation. Fungi reproduce both sexually and asexually via resilient spores, which allow them to spread widely and survive harsh conditions. Asexual reproduction occurs through spore production, fragmentation, broken pieces of hyphae growing into new colonies, or budding in yeasts. Sexual reproduction, often triggered by environmental stress. This involves the fusion of cytoplasm, plasmogamy, and nuclei, karyogamy, to introduce genetic variation. Fungal morphology refers to the physical form and structure of fungi, which range from microscopic single cells to complex, macroscopic fruiting bodies. This structural diversity allows fungi to adapt to different environments and utilize various nutrient sources. Fungi generally fall into three distinct morphological categories. Molds, multicellular fungi made of fine, branching, thread-like filaments called hyphae. A tangled, branching network of these hyphae is known as a mycelium. Yeasts, unicellular fungi that are typically oval or spherical. They reproduce asexually through a process called budding. Dimorphic fungi, fungi that can switch between yeast and mold forms depending on environmental conditions, like temperature or nutrient availability. Unicellular fungi, primarily known as yeasts, are single-celled organisms. Despite being microscopic, they are complex eukaryotes. Their structure consists of a rigid cell wall surrounding a standard eukaryotic internal environment, and they reproduce primarily through a process called budding. Key structural components of unicellular fungi include cell wall, a tough outer layer made of chitin and glucans. This provides structural strength, cell shape, and protects the cell from environmental stress. Plasma membrane, located just inside the cell wall, it regulates the movement of substances in and out. It contains ergosterol, a steroid molecule unique to fungal membranes that stabilizes their structure. Nucleus, a membrane-bound organelle that houses the organism's genetic material, a DNA. Cytoplasm, the gel-like fluid inside the cell that suspends all other organelles. Membrane-bound organelles includes energy-producing mitochondria, the endoplasmic reticulum, Golgi apparatus, and vacuoles used for storage and waste processing. Unlike multicellular fungi, yeasts do not form thread-like structures called hyphae. Instead, they exist as individual spherical or oval cells. In some species, if daughter cells, buds, fail to separate from the parent cell, they can form a chain-like structure known as a pseudohypha. Fungi exhibit a highly specialized eukaryotic structure tailored for nutrient absorption and reproduction. While they range from single-celled yeasts to complex mushrooms, their fundamental body is typically a mesh-like network of microscopic threads. The primary structural components of fungi include 1. Hyphae. The building blocks of multicellular fungi are long, branching, tubular filaments called hyphae. Septate and aseptate are the two primary classifications of fungal hyphae based on the presence of internal cross walls, as we can see in the top right picture. Septate hyphae, divided by cross walls called septa, which have pores allowing cytoplasm, ribosomes, and organelles to flow between cells. Cenocytic, aseptate, hyphae, lack cross walls, functioning as a single, continuous, multinucleated giant cell. 2. Mycelium. When hyphae grow and branch repeatedly, they form a dense, intertwined, root-like network called a mycelium. This web serves as the main vegetative body of the fungus, maximizing the surface area to volume ratio for absorbing nutrients from the environment. 3. Fungal cell walls. Unlike plant cells, fungal cell walls lack cellulose. Instead, they are primarily composed of tough, flexible polysaccharides. Chitin, a strong polymer that provides skeletal strength, the same material found in insect exoskeletons. Glucans, polymers that help form an amorphous matrix within the wall. 4. Fruiting bodies. Sporangium. The visible, reproductive structures of the fungus, e.g., a mushroom cap and stalk. They grow above ground or on the surface of the host to produce and release spores. The morphological classification of fungi categorizes them based on their physical structures, cellular organization, and growth habits. They are primarily divided into 4 main groups: yeasts, yeast-like fungi, molds, and dimorphic fungi. 1. Yeasts. Unicellular organisms that are typically oval or spherical. They lack thread-like structures. Reproduce asexually by a process called budding, forming a smaller daughter cell, or fission. Example include Saccharomyces cerevisiae, brewer's or baker's yeast. 2. Yeast-like fungi. Primarily grow as single-celled yeasts, but can elongate to form chains of connected budding cells known as pseudohyphae, false hyphae. Example include Candida albicans, the fungus responsible for thrush and yeast infections. 3. Molds. Filamentous fungi. Multicellular fungi made up of long, branching, thread-like filaments called hyphae. A dense, tangled network of these hyphae is known as a mycelium. Hyphae structure can be septate, divided by cross walls, or non-septate is cenocytic, a continuous, multinucleated cell. Example include Penicillium and Aspergillus. 4. Dimorphic fungi. Capable of growing in 2 distinct forms depending on environmental factors like temperature or nutrient availability. Typically exist as molds, filamentous, at cooler ambient temperatures, and as yeasts in host tissue at body temperatures. Example, Histoplasma capsulatum, causes histoplasmosis. Fungi reproduce through both asexual and sexual methods, primarily by producing microscopic spores. Asexual reproduction allows for rapid multiplication and identical clones, while sexual reproduction promotes genetic diversity and adaptation. Some fungi can even utilize a parasexual cycle to recombine genetic material without standard sexual phases. 1. Asexual reproduction, anamorph. This is the most common and rapid method of fungal reproduction. It requires only one parent and produces offspring that are genetically identical. Spore formation, sporulation. Fungi produce spores via mitosis. These are typically dispersed by wind, water, or animals. Sporangiospores, formed inside a sac-like structure called a sporangium. Conidia, naked spores produced externally on specialized stalks called conidiophores. Fission, seen in some single-celled fungi, where a mature cell divides evenly into 2 daughter cells through a pinching process. Budding, common in unicellular fungi like yeasts. A small outgrowth, bud, develops on the parent cell, matures, and eventually pinches off. Fragmentation, a piece of the fungal mycelium, the body of the fungus, breaks off and grows into a completely new, independent colony. Sexual reproduction is generally triggered by unfavorable environmental conditions and introduces genetic variation into the population. It happens in 3 distinct stages. Plasmogamy, the fusion of 2 parent cells' cytoplasm, which brings together 2 distinct haploid nuclei, N plus N, into a single cell without immediately fusing the nuclei. Karyogamy, the fusion of the 2 haploid nuclei to form a single, diploid zygote nucleus, 2N. Meiosis. The diploid nucleus divides to produce haploid spores. These spores contain recombined genetic material and are released to grow into new hyphae. Mycoses are fungal infections classified by the depth of tissue penetration in the body and the host's immune status. The four primary types are superficial, cutaneous, subcutaneous, and systemic or opportunistic mycoses. 1. Superficial mycosis infections localized to the outermost, dead layers of the skin, hair, and nails. They cause cosmetic changes, but generally trigger little to no immune or inflammatory response. Examples: tinea versicolor, discolored skin patches, tinea nigra, dark skin spots, and piedra, fungal nodules on hair shafts. 2. Cutaneous mycosis infections that affect the keratinized layers of the skin, hair, and nails, but provoke a distinct host immune response leading to redness, itching, and scaling. Examples: dermatophytoses like athlete's foot, tinea pedis, jock itch, tinea cruris, and ringworm, tinea corporis. Pathogens, often caused by dermatophyte fungi like trichophyton, microsporum, and epidermophyton. 3. Subcutaneous mycosis fungal infections that invade the deeper layers of the skin, subcutaneous tissue, and sometimes bone. They are almost always acquired through traumatic inoculation, e.g., a thorn prick, from soil or vegetation. Examples: sporotrichosis, often called rose gardener's disease, and mycetoma, a localized, destructive infection characterized by swelling and discharging granules. 4. Systemic and opportunistic mycoses, deep fungal infections that affect internal organs, lungs, brain, bloodstream, or become widely disseminated. They are heavily determined by fungal virulence and the host's immune system. True systemic mycoses, caused by dimorphic fungi that can infect healthy hosts. They primarily affect the respiratory tract after spore inhalation. Examples include histoplasmosis and coccidioidomycosis, valley fever. Opportunistic mycoses affect individuals with weakened immune systems, e.g., HIV or AIDS, cancer, diabetes, or hospitalized patients. Examples include candidiasis, aspergillosis, pneumocystis pneumonia, PCP, and mucormycosis. Fungi are essential engines of our ecosystem and human industry. They drive crucial natural processes, like nutrient cycling and decomposition, while yielding massive benefits for medicine, agriculture, and food production. Key useful properties of fungi include medicine and pharmaceuticals. Fungi produce powerful life-saving medications. The discovery of penicillin revolutionized modern medicine, and fungi are still the primary source of key antibiotics, cholesterol-lowering statins, and immunosuppressants. Ecosystem restoration. Bioremediation. Fungi act as nature's recycling cleanup crew. Specialized fungal species, micromediation, can break down toxic pollutants, absorb heavy metals from soil, and even digest certain petroleum-based contaminants. Food and beverage fermentation. Without fungi, culinary staples like bread, beer, and wine would not exist. Yeast is the fungus responsible for fermentation, while other molds create the distinct flavors and textures of cheeses like Roquefort and Camembert. Agriculture and soil health. Mycorrhizal fungi form symbiotic relationships with plant roots. They extend the root network to absorb water and vital nutrients, like phosphorus, while the plant provides carbon in return. Entomopathogenic fungi also act as natural, eco-friendly pesticides, controlling crop pests without toxic chemicals. Food sources. Fungi are highly nutritious dietary staples, providing direct food sources, e.g., mushrooms, truffles, as well as plant-based meat substitutes, mycoproteins, rich in protein and fiber.